are obtaining as part of a blueprint consortium in the work package 4. And I would particularly to emphasize that's a work package presentation, not a personal presentation. So there's a lot of data coming from the collaborating groups in Barcelona, but also Nijmegen and Leipzig and so on. So we deal in this work package with B and T cell malignancies, and I would like to focus today on B cell malignancies. And I would like to do this with a kind of historic slide. We are here in a historic place at the Charité where Virchow worked, but this is a Kiel historic slide where I'm coming from. And actually it's a hand drawing by Professor Lennart, this is this guy, who developed the so-called Kiel classification of lymphomas. And this drawing is showing you the development of a mature B cell from a stem cell over precursor cells, over germinal center cells, at the end to like a mature plasma cell. And though this was drawn up already like in the 1970s, it still holds true. And if we deal with B cell malignancies, we have to deal with a normal development of B cells. The other point I would like to make is, again in this hand drawing, if you have a look into the nuclei, you see the different shape. And this is actually telling us what's the chromatin looking like. So the pathologists recognized this already, that there's a large difference in the B cell development. And actually the person who coined the name chromatin is this guy who worked also in Kiel, this is Fleming, and he also was the first to describe germinal centers. So if we now develop this from a hand drawing more to immunohistochemistry, and we stain, for example, for methylcytosine or hydroxymethylcytosine, and we have a look into a kind of germinal center here, and here's a melanoma metastasis from comparison. You say that see that the staining intensities in the germinal center as compared to the mantle are different, and also the comparison between the different methylcytosine forms is different here. So there's obviously in B cell development a kind of differentiation of a methylation pattern in general. And if you have a look into the expression of DNA methyltransferases, you can see that they are strongly, like DNMT1, expressed in the germinal center, but not in the mantle zone. And this was also shown on RNA level in single cells. So there's clear evidence there is a heterogeneity of the epigenome, at least on the methylation level, in B cells. I would like to talk you today through three different points. The first of them is DNA methylation changes during normal B cell development. And then I would like from this to derive some data from B cell neoplasms. And being a medic running a diagnostic laboratory, I would like to end up with some data on prognostic biomarkers from epigenetics. So if we put the sketch of Professor Lennart in a more modern way, we have a development of a B cell from a progenitor cell over precursor cells, immature cells, naive B cells, germinal center cells, plasma cells. And Marta Kulis in Joaquim Martin Zumbero's laboratory in Barcelona has put a lot of effort to characterize the methylome of these normal B cell subsets. On the one hand using whole bisulfite sequencing, on the other hand on 450K arrays in a subset of cases. And the major finding of the whole genome bisulfite sequencing is that with further development from an immature cell to a mature cell, so here's the immature precursor, here's the mature plasma cell, methylation is lost on a global level. There seems to be a boost of a certain kind of methylation in the germinal center again, but overall there's a loss of methylation. And if you want to group with classes of cells using principal component analysis, you have here the precursor cells, here the naive cells when they go through the germinal center, and you have the plasma cells. One has to be a bit cautious because the precursor cells are from fetuses, and we know that aging is also heavily influencing the methylation pattern. Now, if you use the 450K data and try to classify those cases, those cells, again from the immature to the mature forms, you can see that you can classify the CpGs into modules, and that we have modules which like lose methylation, and we have a few models which gain methylation during the differentiation stage. And having a look into two of these modules, you can see that with module 8, is indicative of losing methylation, and this is particularly affecting heterochromatic regions, whereas polycomb repressed regions gain methylation, and this particularly holds true in the germinal center reaction, so there seems to be a boost of polycomb methylation. If we apply these modules to neoplastic diseases, we have here acute lymphoblastic leukemias derived from precursor cells, CLLs derived from naive or memory cells, diffuse large B-cell lymphomas derived from germinal center cells, and multiple myelomas derived from more plasma cells. Then you can see on the one hand that they have a kind of high polycomb signature. This is what we know, but there's also, with the exception probably of ALL, some changes in the heterochromatic part of the genome. The other point to be made is these are the same changes which occur with age. A hypothesis we heard already this morning could be that it also is telling us something of an age of a cell, because a cancer cell might live longer than a normal cell. This is a kind of background on the normal B-cell development and differentiation. Now let's change to B-cell neoplasms. Within the work package, we are dealing with chronic lymphocytic leukemia, and the data has been published by the Barcelona group already last year. In Germany, we predominantly deal with Burkitt lymphomas, follicular lymphomas, as kind of a German ICGC project. It's a joint effort on mantle cell lymphomas, and there is a poster on multiple myeloma outside, which I briefly would like to summarize, because in this whole genome bisulfite sequencing study by Xavi Aguirre, it's shown that there are two kinds of multiple myeloma, at least based on sequencing. One of them, shown here, has gained methylation. The other has strongly lost methylation as compared to the normal plasma cells. And you can see it here that we have a strong hypomethylation in this multiple myeloma 2, whereas we have a gain of methylation in the multiple myeloma 1. Now, if we have a more detailed look based on 450K arrays, which parts of the genomes are altered, then it immediately strikes you that there is obviously a very high hypermethylation, both in MGUS, which is a kind of precursor of multiple myeloma, and multiple myeloma itself in enhancer regions, such enhancer regions which are obviously playing a role in B cells. We have to say these data come from lymphoblastoid cell lines, which are probably the closest normal cells as compared to plasma cells. So there seems to be a strong enrichment in enhancer regions. And if you have a look why this could be the case, then there is a strong inverse correlation to the expression of a couple of transcription factors, and those to be highlighted likely are PAX5 and IR4. The latter is known to be activated by translocations in multiple myeloma. The former is known to be one of the most prominent transcription factors in plasma cells. So there is a kind of transcription factor shaping of enhancer regions in multiple myeloma. Now, with regard to Burkitt lymphomas, I just show this slide because I think it's the heroic only whole bisulfite sequencing using solid technology. But on the other hand, it gives you a comparison to both technologies and to Illumina sequencing, which is pretty okay. And the third reason I'm showing you this slide is because with this approach, we also have analyzed the integrated EBV epigenome and DNA methylome, and you can see here that we have a strong methylation of the EBV genome integrated into the genome, at least in this Burkitt lymphoma. So this was a pilot study we published earlier this year. Meanwhile, in the German ICGC project, we have profiled like more than 30 B-cell lymphomas and normal B-cell subsets. And what you can see here is that you can nicely apply differentially methylated regions for classification. So we can nicely distinguish Burkitt lymphomas on the left side from follicular lymphomas here on the very right side, and the normal germinal center B cells here in the middle. What you also can see is that obviously the follicular lymphomas, which we know are probably most related to normal germinal center B cells, show a pretty similar profile than the normal germinal center B cells. There are a lot of ways to evaluate this. What we are currently doing in the German ICGC project, we are trying to intersecting these methylation data with the transcriptome and the genomic data we have. And I only show you one example here. This is the SMARCA4 gene, which we know is, and this is a series of more than 800 lymphomas, is highly expressed in Burkitt lymphomas. It's even part of a classifier for Burkitt lymphomas based on gene expression. But though this high transcription might be related to the hypomethylation we see in Burkitt lymphomas, there seems to be a counterbalance on the genomic level because we know that this gene is frequently mutated. So it seems to be on the one hand activated by hypomethylation, on the other hand silenced or at least destroyed by somatic mutations. And there's a nice paper in Nature showing that these mutations we have identified are also functional on the epigenomic level. Now, coming back briefly to discussion we had also earlier this morning, when we want to, at a certain end, apply epigenetics for clinical use, we have to know how stable methylation, for example, is through the course of a disease. And probably the lymphoma in which this can be best studied is the so-called follicular lymphoma, which through mistakes in immunoglobulin recombination typically shows a translocation 1418 activating the BCL2 gene. And we know then through nice studies from Green et al., but also from our group, that there are additional driver mutations like CREPP and like later mutations like MLL2 and others. So we have a clonal evolution, and we know that these patients are frequently relapsing. The other thing we know is that obviously during high-grade transformation, during progression, we get a more diffuse pattern of a methylation if we have a look into this, into global issues. So what we did actually, we tried to profile the methylation in single steps of the development of this follicular lymphoma. So the very early step is a so-called in situ follicular lymphoma, where you have also only tumor cells in like these germinal centers, which are neoplastic. So we microdissected those, compared these to full-blown lymphomas of the same patient. And what we can show is that at least taking polycomb targets as methylation marks, we see an increase of methylation from the initial step, so the in situ follicular lymphoma, to the later step, which is a full-blown lymphoma. So what we can say is that at least at like the second step, methylation is already playing a role. We can in more detail characterize this by deep sequencing of such polycomb targets again, in which we can draw this kind of pedigrees and can show how the evolution from a predominant clone, epigenetic clone here in the relapse is changing to a more altered clone. And the third analysis we have done, we have, together with a group in Leipzig, we have characterized a series of cases in which we had initial diagnostic materials and relapse materials, and we built phylogenetic pedigrees based on the IGHV mutation analysis, and this is shown here. The initial diagnostic sample is the red one, the relapse is the blue one. You can see there are patients which hardly have any evolution, and those show the same methylation profile. And we have patients which have a clear evolution of a distinct clone or subsequent evolution. These show alterations. And if we put all this together, we come to the conclusion that obviously the genomic evolution goes along or is parallel by an epigenomic evolution, again, focused on polycomb target genes. So if we want to identify biomarkers, it's probably not the wisest decision to take these polycomb targets because we know that they further evolve during the evolution of a disease. So this brings me to my final part, DNA methylation changes as diagnostic or prognostic markers, and I would like to introduce you into two studies briefly. The first one is dealing with diffuse large B-cell lymphomas, and it's actually going back to a paper we published back in 2009, in which in the unpublished part we could show that the more methylated the tumors are, the inferior the prognosis. So you can see this survival curves do not really look reliable. But we took this as an initial hint that we could probably identify prognostic markers there. So we selected those most highly informative CPGs. And in addition, we took those CPGs published in the literature as being prognostic, either with a favorable prognosis or an unfavorable prognosis if methylated. And then using these data, we profiled a large German prospective randomized trial. Actually, we could get DNA from FFPE materials from more than 400 patients from this trial, allowing a lot of multivariate analysis. I would only like to show you the major findings here. The first conclusion was all these markers we identified and those which are published before, which we tested based on bisulfite pyrosequencing, did not turn out to be prognostic if you apply them to a prospective trial. The only exception was the methylation of a P16 gene, which regarding event-free survival and also overall survival was prognostic. We then evaluated this further in a multivariate analysis, taking into account all clinical variables, and still the methylation has is an indicator of inferior prognosis. So from all the marks tested, only one finally standed the rigorous clinical evaluation in a clinical trial. The other example I would like to show is again from the Barcelona group, which last year published the data on CLL, and as part of the study they did 450K arrays and could show that they could classify based on methylation the CLLs into a naive B-cell-like CLL group, into a memory B-cell-like CLL group, into an intermediate group. And this is the overall survival, and you can see that the memory B-cell-like group had a much favorable prognosis than the naive B-cell-like group. So this was an initial evaluation study, and this prompted us in a joint effort to have a more detailed look into this. Now you do not want to make a 450K array on each patient. So the idea was to bring down the informative CPGs to a set of 5 CPGs which can be tested by bisulfite pyrosequencing. And obviously there are various of such combinations. We ended up with one. And the correlation between the array and the bisulfite pyrosequencing for these 5 CPGs and for the classification was perfect. And if you have a look then into the clinical data, then is here the training series from Spain, and you can see, have a look only into the survival curves, that they nicely discriminate into the 3 groups as shown in the initial study. But probably more important is this is a cohort which is enriched for quite favorable patients. Together with a group of Martin Dyer and Lester, we had a cohort with more unfavorable patients. And what you can see, despite that the clinical factors were more unfavorable, we again can nicely distinguish the prognostic groups based on the bisulfite pyrosequencing of the 5 markers alone. Now, the ultimate proof is obviously for the prognosis, the multivariate analysis, including all the clinical data, like Binet stage or the CD38 expression. And what you can see here from the analysis is that the epigenetic subgroups still have a high discriminatory power. And in the Cox regression model, the epigenetic classification was the strongest predictor of outcome in CLL. Now, if you want to apply this in a diagnostic setting through different laboratories, you have to test it in different laboratories, and this is actually the final part of the study we did. We took the same sample from the same patient, divided them into one part of the cells analyzed in Barcelona, the other in Kiel. We did all the preparation more or less exactly the same. And we ended up with a close to perfect agreement with a kappa of about 0.9. And you can see here the results for the different CPGs. So we can prove a nice interlaboratory concordance between these marks. So we are pretty sure this is a quite validated marker for prognosis in CLL. So let me summarize. I hope I could show you that there is a dynamic evolution of methylation in B cells. We know that there are changes in B cell neoplasms, and I have to say, from what we can see so far, the changes might differ between the different lymphoma entities. And finally, there might be some of the markers which we can use for diagnosis and prognostication. And that's actually the teams which have done most of the work. The CLL team from Barcelona, the German ICGC project funded by BMBF here, and those who did the predominant or the major work is Iñaki Martin Sobero's group and Anke Bergmann, Ola Amarpohl from my group from the both teams in Barcelona and Kiel. Thank you very much.
Thank you very much, Reiner.
I was just wondering, when you're comparing with the lymphoma with the, I guess, the initial and then the relapse samples, how much do you think the change in methylation is a generic part of progression, or do you think it's related to the type of treatment that the patient received?
Obviously, this was one of the questions we addressed. Treatment, we do not have evidence that it is associated with treatment. We do not have evidence that it is associated with time. So you would probably expect that if there is a longer time to relapse, you might have a more pronounced change. This doesn't happen. That's the reason why we think there is a kind of active mechanism. And I haven't shown you the data, but if you compare the somatic mutations in chromatin genes, for example, with the methylation patterns, you identify CRAPI-P as one of the genes. If mutations are there, it might shape the methylation status. And this fits nicely with a Green study who also identified CRAPI-P as like the second hit after the 1418.
I see no further questions. Then thanks again for all the speakers.